Welcome back to The Deep Dive, the show where we take the operational blueprints of complex systems, and today it's the human body, and really extract the core insights you need to understand how things actually work.
Today, we are peering behind the curtain at the ultimate silent partner. I mean, this is the command center that manages your entire physiological existence, completely outside the realm of consciousness.
We're diving into the autonomic nervous system.
The NNS, yeah.
I love that framing, because if you stop thinking, or, even if you were completely unconscious, your heart keeps beating, you keep breathing.
Digestion continues, blood pressure stabilizes.
Exactly. The ANS is that sleeping pilot maintaining the plane while you just rest.
Precisely. And our mission today is to systematically walk through the structure of this unconscious controller. We're going to compare its two major subdivisions, the sympathetic and the parasympathetic.
And break down the anatomy and the chemistry that dictates your body's response.
Whether you're facing a crisis or, just enjoying a quiet meal, the breadth of what the ANS manages is just staggering.
Yeah.
It coordinates every major internal system.
Cardiovascular, respiratory, digestive, even reproductive functions.
And it also handles the subtle routine adjustments. Things like making sure your body fluid concentrations, your water, electrolytes, all that stuff.
Stays at the perfect level.
It truly is the unsung hero of homeostasis.
Okay, so let's start by contrasting it with the system we can control.
The somatic nervous system, or the SNS. That's the one that moves your skeletal muscles.
And the distinction is really structural, isn't it? When I choose to move my arm, that signal from the CNS goes straight to the muscle. It's a single lower motor neuron.
Correct input, direct control, exactly. But the ANS, which controls all the things you can't consciously manage,
The visceral effectors.
it requires a major detour. And this is the hallmark two-neuron chain.
Okay.
So the first neuron, the visceral motor neuron, it starts in the CNS, and its axon is what we call the preganglionic fiber.
And that preganglionic fiber doesn't go all the way to the organ itself. It stops at a peripheral collection of neurons.
An autonomic ganglion.
Right.
Correct.
Yeah.
That ganglion is like the switchboard. The preganglionic fiber synapses there onto a second neuron and the axon of that second neuron.
The postganglionic fiber.
That's the one that finally travels to the target organ, whether it's cardiac muscle, smooth muscle, a gland, whatever.
So the ANS is inherently a more distributed system. It seems like this two-step process allows for much broader regulation.
It does. And it also differentiates the sensory inputs. I mean, while the SNS is driven by our external senses, the ANS relies on information from internal visceral receptors.
Sensors monitoring things like the pressure in your bladder or the stretch in your stomach.
Exactly.
Okay, now that we have the structure, let's introduce the two famous opposing forces.
First, the sympathetic division. It's also known as the thoracolumbar division because of where it originates anatomically.
This is the famous fight-or-flight system.
That's the one. It's active during periods of, exertion, stress, or emergency. Its entire job is to stimulate metabolism, increase energy, and ramp up your alertness.
And the system that brings everything back down.
That's the parasympathetic division, the craniosacral division. This is your rest and repose, or sometimes rest and digest system.
Right.
It activates under resting conditions. It prioritizes conserving energy and promoting sedentary activities like, digestion. Most of the time, these two have opposite effects.
They're antagonistic.
Precisely.
But before we dive into the anatomy of a crisis, we have to mention the third, often overlooked division.
Ah, yes, the enteric nervous system, the ENS.
Yeah.
What's fascinating here is that the ENS is this extensive network of neurons embedded entirely in the walls of the digestive tract.
I read it has something like 100 million neurons.
It's equivalent to the entire spinal cord. And its main job is to coordinate complex local visceral reflexes, mostly without needing any direct input from the brain. It's a nervous system within a nervous system.
Wild. Okay, let's focus on the sympathetic division. If its job is a widespread rapid response, the structure must be designed for, maximum reach and speed.
Absolutely. And the structural organization really facilitates that widespread reaction. The preganglionic neurons are strictly limited to the lateral gray horns of the spinal cord, specifically segments T1 through L2.
Which is why we call it thoracolumbar.
Exactly.
So all the input comes from that central trunk, but the effect has to reach your head, your limbs, all your organs. How does the signal get that incredible reach?
Through divergence and distribution. A single preganglionic fiber can synapse on up to, 32 ganglionic neurons. It just spreads the signal massively.
Wow.
And the fibers can use 3 different types of switchboards, or ganglia. The first and most extensive path is the sympathetic chain ganglia.
also called paravertebral ganglia.
Yes, because they run like a chain right alongside the vertebral column.
Hmm.
Preganglionic fibers enter this chain. They can travel up or down within it, and then synapse.
And this chain runs the entire length of the spine, even though the input only comes from T1 to L2.
It does, and this allows postganglionic fibers to reach peripheral targets everywhere, in the head, neck, limbs, body wall. Things like sweat glands or the muscles that make your hair stand on end.
So how do the fibers get in and out of that chain?
So, when the myelinated preganglionic fiber enters the chain, it uses the white ramus communicants. Then, when the unmyelinated postganglionic fiber leaves to go to the spinal nerve, it uses the gray ramus communicants.
White because it's myelinated, gray because it isn't.
Exactly. It's an architecture that ensures targets far above or below that T1L2 region still get sympathetic signals.
Okay, that's the first path for the body wall and limbs. What's the second route, the one for the abdominal organs?
Yeah, it would be the collateral ganglia or prevertebral ganglia. They sit anterior to the vertebral column. For these targets, the preganglionic fibers they bypass the chain ganglia entirely.
And they converge to form what are called splanchnic nerves.
Right, so this is like a separate, dedicated command route for the really vital internal organs.
Like a direct line.
A direct line. There are three major stations here. The celiac ganglion mostly handles the stomach, liver, pancreas. The superior mesenteric ganglion takes the small intestine and the first part of the large intestine.
Not the third.
The inferior mesenteric ganglion. It manages the end of the large intestine, the kidney, bladder, reproductive organs.
And that brings us to the third destination, the one famous for that massive whole-body chemical response.
The suprarenal anatomy. At the center of the adrenal gland, these are essentially modified sympathetic ganglia.
So preganglionic fibers go there.
And synapse directly onto specialized endocrine cells. And instead of releasing a neurotransmitter onto a muscle, these cells just dump massive amounts of epinephrine and norepinephrine directly into your bloodstream.
To act as hormones.
White thread hormones, yeah.
This anatomical arrangement, especially with the adrenal gland, really explains why that sympathetic response feels so dramatic and body-wide. When the system activates, what happens functionally?
It's a coordinated preparation for extreme physical demands. You get increased alertness, often euphoria, that feeling of being on edge or even ignoring pain.
Right.
Your cardiovascular activity just spikes. Heart rate and blood pressure soar. Breathing rate increases.
And the energy mobilization must be key.
Oh, absolutely vital. The body taps into its savings account. It accelerates glycogen breakdown in the muscle and liver, releases lipids from fat tissue.
All available energy is made immediately accessible.
Instantly.
Let's talk chemistry. The communication chain has two steps, but really only two main chemicals, right?
For the most part. All sympathetic preganglionic neurons use acetylcholine, or HA, and this synapse is always excitatory.
But the output after the ganglion, that's different.
The output is mostly norepinephrine, or NE, so most postganglionic fibers are adrenergic. But here's an important nuance. A few postganglionic fibers still release HA.
Really? Where?
Specifically the ones that go to your sweat glands and the blood vessels in your skeletal muscles.
So even within the sympathetic system, there's some chemical variety. But what about the adrenal hormones? Why are they so much more powerful than just a synaptic release?
It's about persistence. NE and E released into the bloodstream circulate as hormones. And because the bloodstream doesn't have the rapid breakdown enzymes you find at a synapse.
The effect lasts longer.
For several minutes. And they affect every cell in the body that has the right receptor, ensuring the response lasts long after the immediate crisis is over.
And that brings us to receptor diversity. We have alpha receptors and beta receptors. Why have two different types for the same chemical signals?
This is how the system fine-tunes the crisis response. I mean, imagine you need to speed up the heart, which is a beta receptor thing, while at the same time slowing down digestion, which is more of an alpha receptor thing on digestive muscle.
So the same chemical can have different, even opposite effects.
Exactly. It all depends on which type of receptors on the target cell. NE primarily stimulates alpha receptors, while epinephrine stimulates both, which is part of why that adrenal output is so effective.
That makes perfect sense. To ground this in reality, let's look at what happens when this system breaks, like in Raynaud's phenomenon.
Raynaud's is a great clinical example. It's an excessive, sort of pathological sympathetic response, usually to cold or stress. The sympathetic output causes extreme vasoconstriction in the periphery.
In the fingers and toes.
The blood vessels just clamp down too hard, leading to temporary loss of circulation. The skin might turn pale or blue. It's the fight-or-flight system just overreacting.
And what about the localized breakdown in the face, Horner's syndrome?
That's often caused by unilateral damage to the sympathetic chain in the neck. The result is a loss of sympathetic innervation to one side of the face. You get pupil constriction, a droopy eyelid, and flushed skin because the blood vessels are just permanently relaxed.
Okay, now let's shift gears. The parasympathetic division. If the sympathetic system is an emergency broadcast power with massive reach,
Then the parasympathetic is a highly precise, localized landline.
and this localization is reflected in its origin, the craniosacral origin.
Yes. From the brainstem associated with cranial nerves, 3, 7, 9, and the mighty 10, and then from the sacral spinal cord, segments S2 through S4.
And unlike the sympathetic system, where the ganglia are far from the target, here they're right next to or even inside the organ.
Exactly. We call them terminal ganglia if they're very close, or intramural ganglia if they're literally within the tissue wall of the organ.
And this close proximity forces the effects to be specific and local.
It has to be.
I remember the sympathetic divergence was huge, up to 1 to 32.
What about the parasympathetic system?
The difference is striking. A typical parasympathetic preganglionic fiber only synapses on maybe 6 to 8 ganglionic neurons, and they're all focused on the same target organ.
So that low divergence is the key to why the rest and repose effects are so specific.
It's the primary reason. You don't activate the whole body at once.
But wait, if the input is so localized, how does it control so much of the torso and abdomen? That must be the vagus nerve doing all the heavy lifting. You've hit on the massive strength and vulnerability of this system. The vagus nerve, NX, provides roughly 75% of all parasympathetic outflow.
75%.
Wow. It travels through the thoracic cavity and down into the abdominopelvic cavity, regulating the heart, lungs, liver, digestive tract, all the way to the last segments of the large intestine.
So if something disrupts the vagus nerve, a tumor, an injury, you effectively lose 3/4 of your ability to settle down and digest. That sounds like a serious systemic vulnerability.
It's a critical hub. The remaining parasympathetic control for the very lowest regions, the pelvic organs, that comes from the sacral outflow. Fibers from S2 to S4 form the pelvic nerves there.
Okay, so the general function of this system is relaxation, conservation, and processing. Everything related to getting energy in and waste out.
Correct. We see constriction of the pupils, which helps you focus on nearby objects, massive secretion by digestive glands, salivary, gastric, pancreas, and ramped up smooth muscle activity along the digestive tract.
And at the same time, heart rate and force of contraction go down.
It's the ultimate energy-saving mode.
And when we turn to the chemistry, the simplicity is, it's a welcome contrast to the sympathetic system.
It is remarkably straightforward. All parasympathetic neurons, preganglionic and postganglionic, are cholinergic. They all release ACA.
And that leads directly to the brief, highly localized effects, right?
Absolutely. The ACA that's released is immediately and rapidly inactivated by acetylcholine esterase right at the synapse. So the signal hits the target, gets the job done quickly, and then just vanishes.
Ready for the next precise command.
Exactly.
Now, despite having only one neurotransmitter, AC, there are 2 crucial receptor types. This can be confusing. How do we differentiate nicotinic receptors from muscarinic receptors?
Just think of them as 2 different kinds of locks. They're both opened by the same key, which is key A, but they initiate 2 completely different responses inside the cell.
Okay, tell us about the first lock, the nicotinic receptor.
Nicotinic receptors are found everywhere. There's a synapse between the 2 neurons, so on all ganglionic neurons, both sympathetic and parasympathetic.
On both sides.
On both sides. And when AC binds here, the effect is always excitatory. It just passes the signal on.
And the muscarinic receptor, the second lock.
Muscarinic receptors are found at the actual neuroeffector junctions of the parasympathetic system, the connection point to the target organ. And here, when AC binds, the effect can be excitatory or inhibitory,
Depending on the target cell.
entirely depending on the enzyme pathways inside that target cell. This is critical for controlling things like heart rate, which is an inhibition, versus digestive muscle contraction, which is excitation.
The reality then is that these 2 systems rarely operate in isolation. Most vital organs are under dual innervation.
Meaning they receive instructions from both.
And where that exists, the effects are usually antagonistic. They're in a constant push-pull for control over things like your heart rate.
And this push-pull dynamic means they have to physically intermingle. Sympathetic postganglionic fibers and parasympathetic preganglionic fibers. They sort of weave together to form these intricate nerve networks called plexuses.
These plexuses sound like massive anatomical intersections.
They are. In the chest, You have the cardiac, pulmonary, and esophageal plexuses. In the abdomen, you have the mighty celiac plexus, the inferior mesenteric plexus, and the hypogastric plexus. These dense networks ensure that both crisis commands and rest commands are available simultaneously to the major organs.
Finally, let's talk about the functional units of the ANS, the visceral reflexes. These are the automatic motor responses that run everything.
We categorize them by where the processing happens. The long reflexes are the systemic ones. Sensory input goes to the CNS. It's processed in the spinal cord or brainstem, and then motor commands are carried out by the ANS.
These coordinate the activities of the entire organ system.
Yes, but the truly autonomous actions are the short reflexes.
They're magnificent because they bypass the CNS entirely. The processing happens locally within sensory neurons and interneurons located inside the autonomic ganglia themselves.
Like in the enteric nervous system.
Exactly. The ENS runs largely on short reflexes. They manage very simple, localized motor responses, ensuring this fine-tuned control without bothering the brain. A perfect capstone example for all this is the urinary reflex, which requires both systems to cooperate and conflict.
It's a great illustration. The urge to urinate is driven by the parasympathetic nerves from S2 to S4. They cause the bladder wall to contract and relax the involuntary sphincter.
That's the go signal.
That's the go signal. But the sympathetic nerves from L1 and L2 do the opposite. They inhibit that bladder contraction and contract the sphincter, which promotes urine retention.
So holding it in is a sympathetic fight or flight response, while letting go is a parasympathetic rest and digest action.
Precisely. And you can see how critical this is when there's an injury. If you have a sacral spinal cord injury that cuts off that S2-S4 parasympathetic outflow, you get a condition called detrusor areflexia.
An autonomous bladder.
Yes. And without that parasympathetic signal, the bladder can't reflexively contract and empty. It leads to chronic retention and overflow incontinence. It just proves how dependent these complex processes are on that intact ANS circuitry.
Wow. Okay, so to reinforce the core takeaways of this deep dive, let's just summarize the blueprint.
If you're comparing the two, remember this. The sympathetic system has short preganglionic fibers. It's highly divergent, mostly NE-driven, and it's designed for a widespread hormonal, minutes-long crisis response.
And the parasympathetic.
Long preganglionic fibers, highly localized, entirely H-driven, and designed for precise, rapid, short-lived rest and repose actions.
This entire deep dive has been about recognizing the extraordinary degree of control exerted by processes entirely beyond our conscious awareness.
And that leads to the final provocative thought. Consider the sheer computational power that is housed in the short reflexes of the enteric nervous system. While your conscious brain is distracted, millions of local neurons within your gut wall are coordinating the entire complex biomechanics of nutrient absorption, motility, and secretion.
A nervous system within a nervous system.
Exactly. It means that these enormous, complex physiological functions can operate almost entirely autonomously, requiring virtually no oversight from central command and demanding only that you supply the raw materials.
A powerful reminder that our bodies are perpetually functioning on autopilot. Thank you for joining us for this deep dive into the anatomy of the unconscious controller.